سبحانك لا علم لنا إلا ما علمتنا إنك أنت العليم الحكيم رب اشرح لي صدري ويسر لي أمري واحلل عقدة من لساني يفقهوا قولي نويت تعلم وتعليم والنفع والانتفاع والإفادة والاستفادة والحث على التمسك بكتاب الله وبسنة رسوله صلى الله عليه وسلم وابتغاء وجه الله ومرضاته وقربه وثوابه أما بعد. Respected students, can you hear me?
Yes, Doctor.
Yes, Doctor.
All right, Alhamdulillah. Did you have a class this morning?
Cancel.
Cancel. Okay. We could have started earlier. Okay. Anyway, I am not able to have presenters window when presenting this. I hope you can see my face as well as the slide. Yeah?
Yes.
Yes.
Okay.
Yes.
But I cannot see you guys. I will try again to share the full view of the slide. Tell me if you can see it now. Hilang ke atau it is there?
It is there but not full.
Not full.
Yes.
What can you see? What does it say?
The importance of immunity.
Okay, if I move the slide, can you see it's moving?
No, it doesn't move.
No.
Okay, something is wrong. Okay, I will keep it like this so you can see on my left all the slides number 21, 22, etc. Yeah?
Yes.
Is that correct? Okay. So if I move the slide, you can see it moving.
Yes. Yes.
All right. So we will start with part 2 of the lecture on the importance of immunity. Yeah. So last week we have introduced to you about the concept of immunity, the concept of self and non-self. Yeah, okay. So trying to change the perspective of the immune system as only a defense system. Okay, we want to change that. Okay, because the immune system is not just about defending us from invading agents. It is about also about maintaining health. Okay, so in cases like allergic reaction, hypersensitivity, some of this does not involve any invading agents or pathogens, yeah, but rather it involves how our immune system communicate and interact with our own body. So if the ability to recognize self is broken down, then we have problem. We will have diseases, okay, reactions, which is not supposed to happen. If you look at the list of assignment that I have given, I hope you have already chosen your topic. Yeah, if you have not been told by Farah Nasirah, I have already released the topics for the group assignment. Yeah, you need to do group presentation. So in one of the topics, I have put the use of the antibodies as a diagnostic tool. Okay, so again trying to impart the understanding that we can use the immune system for other purposes. Okay, not only about defending ourselves from invading agent, not only about maintaining the equilibrium and the intended interaction between the immune system and the body, but also for purpose like diagnostic. Okay, so we hope to have a nice presentation for that part. Okay, that part I can't cover under the basic immunology. So we have a window an opportunity to appreciate the immune system from that perspective. So, and then we also showed to you some one video on Chacha and also some statistics on the effect of vaccination. Okay, so vaccination is one of the main and major practice, the sexual organs, etc. So the immune system is also of importance. It actually link many of the systems in the body. So we will revisit about the need for having a good immune system. If we ponder about our daily life, the human actually represents a very secure home and a source of very nutritious meal for any organisms that can overcome our defenses. That's why we have this invasion by the organism taking place every minute, every seconds. In fact, we are already infested by many of these organisms. Yeah? If we take a swab of our skin, a swab of our nose, there are already lots of organisms which stays there. Yeah, they are not invading because they are not they are existing in the commensalism mechanism. Okay, manner. They are not invading, meaning that they are not causing us any problem at the moment. But some of them are opportunistic. Anytime that the immune system is low or lowered, like when we are not taking enough meal or when we are undergoing chemotherapy, so they can overcome these defenses. Yeah, these organisms, they could be viruses, okay, like COVID-19. COVID-19 is a type of virus, and then it could be bacteria. Okay, bacteria could be an anaerobic bacteria or an aerobic bacteria. The difference between the two is whether they need the presence of oxygen to survive or not. If you look into, everyone's mouth, yeah, the oral cavity is actually full of bacteria. Okay, I wouldn't say virus, but bacteria. Yeah, it is estimated that about 90%, almost everyone, yeah, of us will have some degree of gingivitis, yeah, inflammation of the gingival tissue, um, which is caused by caries, yeah, tooth caries, which is um, due to bacteria forming plaque on the teeth because of the accumulation of food that happen on the teeth, so they can grow on that area of teeth. So some of these gingivitis can deteriorate into periodontitis. Yeah, periodontitis is a much deeper infection which involve also the periodontal ligament and can go as far as into the mandibular muscles and bone. So in the periodontitis, the depth of the bacterial infection is much deeper. Okay, actually we can classify the periodontitis. You can have a look, you can read about periodontitis later on. We can classify them into 3 levels. Okay, the 3 levels. So we have the teeth. So the infection get into the space between the teeth and the gingival lining. So it creates a pocket. Okay, this pocket is full of bacteria. It then attacks both the tooth and as well as the gingival lining. Yeah. So if we take this pocket out, there are 3 phases. The topmost is mostly aerobic bacteria because they need oxygen. The second level is a mixture of anaerobic and aerobic bacteria. Okay, but the bottommost is predominantly anaerobic bacteria. Okay, so they are already there in our body or on our body. Yeah, so if you go to the GI tract, there's a lot more bacteria there. Some of them are useful bacteria, some of them are opportunistic bacteria, and some of them are bacteria that is trying to invade by being attacked by our system. Fungi is another very important organism. On a normal daily basis, we don't see fungi much anywhere in our body, but some of you may have some fungi on the sole of your foot. Yeah, apa tu? And then also some of you, especially ladies, you have to be very careful because down below, your parts down below is most of the time moist, and this can give a very good area for fungi infection. Then we have protozoa and worms, and these are all very abundant in our environment. Yeah, abundant in our environment. And they also have very short generation times. Some of them can produce so many replicates within 1 hour. Not only that, so they enlarge in number in very short time. They also evolve very rapidly. Yeah, evolve very rapidly. And by evolution here, we mean that the change in terms of their protein expression and change in terms of their genetic expression could happen very, very fast, such that the immune system is unable to catch up. Yeah, the immune system unable to catch up, and then when we make out some medication, they also rapidly phase out because they can no longer attack the old generation of the virus or the bacteria. And this is also what we are currently worried about. We can still have a very effective vaccine or not. Yeah. So since the first wave, we have seen there were Wuhan type, and then the UK type, the South African type, and very recently we have the Nigerian variant of the COVID-19. So a very big question now is whether you take the AstraZeneca vaccine, the Sinovac or the Pfizer, whether they can still be effective come what not, yeah, come whatever type of variant of COVID-19, whether they can still be effective against the new variants. So fingers crossed, the one that we have now would be able to identify, to help us identify and stimulate the immune system to attack any type of variant. So there are other infections other than COVID-19, like the flu, for example. Yeah, we also have a very rapid evolution, and this is why even though we have already received one infection, flu infection, we can still have flu infection again and again and again, yeah, because of the huge and rapid evolution in terms of their protein expression and genetic expression. So the immune system is important because we are already attacked. We are being, very, without them, we are easy target for lots of organisms. Yeah, thousands of them. If we are talking about the parodontitis just now, within one small pocket, one tooth, we could identify up to 100 types of bacteria. Yeah, the anaerobic and the aerobic bacteria, about 100 within one small pocket. So if we take into account the rest of the body, we could have thousands, yeah, hundreds of thousands perhaps of different type of bacteria. So we are currently living or appear to be living healthy because of the immune system. Yeah, despite these myriads of organisms staying within ourselves, on our body. We appear healthy because of the immune system. Okay, and we also move. We are very mobile. Humans are very mobile. We go from here to another place all the time. Yeah, and we eat all sort of food. Yeah, we have vegetables, meat, even water that we get in, that we drink. These are full of germs, full of organisms actually. So the area of contact with the environment are huge. Yeah, we have skin. Skin is the largest organs in our body. So we are interacting with air. We are also interacting with surfaces. Okay, and the only barrier is the skin. When we take air, okay, through our nose, then to the respiratory system, all this respiratory tract, they are in contact with the organism in air. Okay, so COVID-19, for example, could be transmitted through air, through aerosols. That's why we are wearing masks. We are wearing masks not because we want to prevent the organism from coming in, but actually, if we are infected by COVID-19, we want to prevent when we breathe or we bersin macam tu, the COVID-19 virus does not goes to another person. Yeah, so 1 meter is the safe distance if we bersin or we cough. Okay, so that is the safe distance. So respiratory tract, gastrointestinal tract, because we may have eaten ulam, very raw material. Some ulam they are growing very close to the ground, macam pegaga, like pegaga is very close to the ground. Some ulam like perhaps kemangi or selasih, they are taller, so we can pick the shoots, and we don't actually need to wash much. Yeah, we just need to bilas macam tu je. Just air lalu should be okay. But pegaga is better if we rendam the leaves. Yeah, put it inside a bowl of water and leave it there for a few minutes before we take the pegaga. Yeah, it must be washed thoroughly. So if not, then all sort of organisms that were coming from the soil could still be there. Okay, so gastrointestinal tract, and then urogenital tract. Of course, when you become at the age of what we call the sexually active age, yeah, when you get married, etc., the contact and the risk of infection through the urogenital tract will be increased. Okay, and we also, when we have interaction with the surfaces, okay, actually we have wounds all the times in our on our body. Yeah, there are cracks. If you look at your sole, yeah, there may be cracks on your skin. You may cut yourself while you were in the kitchen. So all this increase the areas of contact with the environment. So we have the surfaces like skin to protect us against the invasion. So the skins and all the epithelial linings on the respiratory tract, the GI tract, urogenital tract, etc. These are all also part of the immune system. So there is another perspective that we want to change, or we want to correct, that the immune system is not just about antibody, it's also about the epithelial lining. Yeah, we will look at the innate system and the non-innate system, the adaptive system, later on in next week's lecture. Okay, so the innate system is one of the components of the immune system. Yeah, and then we have the cellular or the adaptive components. So all these immune cells, they originate from the bone marrow. And from the bone marrow, they then migrate to the peripheral tissues. When they are in the bone marrow, they need to be stimulated to grow and then to proliferate, making more numbers, and then to differentiate, to have more specialized function. But they will not become mature yet. They need to migrate, some of them. They need to migrate. If the T cells, for example, they need to migrate to the thymus and mature there. The B cells, they stay and mature in the bone marrow, but to carry out their real function, they still need to migrate to the peripheral tissues. And to do the migration, they need to be in the blood. So they will need to be released into the blood. Some will be circulating, some will be residing in certain organs. Some also will be circulating in specialized vessels, which we call the lymphatic system. And during the circulation, there will be tonic signals. Tonic signal means signals that is making the cells alive, making the cells continue to have and to be able to carry out their functions. So it's stimulatory signals. So this will be received by the receptors on the cells, and then to ensure survival of the immune cells for the length of time that is needed. Okay, and at one particular time, all of these cells will be signals that the body receives saying that these cells are already old, they cannot be maintained, so they will need to be destroyed. And then the cycle goes on. So what are the cells of the immune system? Okay, we have mentioned that these cells come from the bone marrow cells, what we call the pluripotent hematopoietic stem cells. So in our body, the bone marrow contains the most numbers of stem cells, and they are hematopoietic, which means that they give rise to the blood system cells and the immune system cells. So why was it called hematopoietic stem cells? This is due to the history, because in the beginning, we could only appreciate that this population of stem cell could give rise to the hemato or the blood cells. It was later on that we then also be the immune cells. So the terminology remains as pluripotent hematopoietic stem cell. So from this original population of cells, certain signals acted on them, so that within the bone marrow, they then diverge. Some will become or follow the lymphoid pathway, some will follow the myeloid pathway. Okay, so if you look at this slide, those on the right, in red, these will become the common myeloid progenitor cells. Okay, common myeloid progenitor cell. Progenitor means the early cells. Okay, the progeny. So they will evolve and differentiate to become other type of cells. Some of them will become the granulocytes and macrophage progenitor cells. Some will become megakaryocyte or erythrocyte progenitor cells. Okay, there are tonic signals that maintain this division. This division is more or less random. Some will become the macrophage progenitor, some will become the erythrocyte progenitor. So, and then once they are in that particular pathway, there are tonic signals that will maintain them in that particular pathway. They are not able to revert or go back, to become the myeloid progenitor cells. Okay, so there is, they are unable to revert. But in recent years, under the regenerative medicine, we try to find ways in which we can allow these cells that have some differentiated function to go back along the pathway towards the myeloid progenitor cells. But there is another story. Okay, so if we follow this common myeloid progenitor, they become the granulocyte and macrophage progenitor. Some also will become the megakaryocyte, erythrocyte progenitor. So if we look at the megakaryocyte, erythrocyte progenitor at the very end on the right side, they become the megakaryocyte as well as the erythroblasts. Okay, the blast in any cell, if you will come across this terminology with the word blast at the end, this means that it is still very immature. After the blast phase, then the cell will reach the site phase. Yeah, so they become the erythrocyte. Yeah, this is a more functional and more differentiated. We can sort of say this is the final phase of the cell. So from the erythroblast, they become the erythrocytes. Likewise, the megakaryocyte, this is already the final phase. They will not evolve anymore. They just being split. Yeah, the nucleus has split, and they become the platelets. Okay, so or final name for it is the thrombocytes. The granulocyte macrophage progenitor in the middle, okay, they will, as their name suggests, become the granulocytes, the granulocytes, or also we call them polymorphonuclear leukocytes. Okay, why the name is so? Okay, polymorphonuclear means that the nucleus has many form. Yeah, many form. And they have nodes. They are not single shape. They are not well rounded. Yeah, they form nodes. So some of them, like the neutrophil, they have 3 to 6 nodes. And also the name granulocytes. This denotes the fact that these cells have granules in their cytoplasm. Yeah, if you look at the neutrophil, there are a lot of granules outside the nucleus. Likewise, the eosinophil and also the basophils. The mast cell also have a lot of granulocytes. And then we have the monocyte, okay, which together with the mast cells, okay, they become the mast cells as well as the macrophage. You can see the granules forming in the final form of the cells. Now, the granulocytes, okay, they also contribute to the formation of the immature dendritic cells. Okay, on the very left side of the slide, okay, we have the lymphocytes represented by the B cell, T cell, as well as the NK cells. The lymphocytes, or the cells that form the lymphoid progenitor cells, are also thought to be able to become the immature dendritic cell. So for the dendritic cells, there is a khilaf ulama there. We are unsure of the original pool of cells, whether they are from the lymphoid progenitor or not. Yeah. So the current body of evidence is saying that it can come from both progenitor cells. So the immature dendritic cells then become the mature dendritic cells. Okay. Within the tissue. So they are released from the bone marrow, they get into the blood in their immature form, and then they get into the tissue where they will become mature there. Okay. The lymphocytes, yeah, the B cell, T cells, and the NK cells. Okay, the NK and the T cells especially, they need to migrate to the lymph nodes. Okay, to the thymus before they become mature. The B cells, they remain in the bone marrow, mature, and then being released in the blood. They then get into tissues and organs in our body, chief among these is the lymph nodes. So lymph node is all over the body. If you notice, when we have infection or fever, some of our lymph nodes will become enlarged. Okay, this is because the B cells and the T cells residing in the lymph nodes, they will be in contact with antigens from the invading organisms, and they will be stimulated uh, and differentiate. The B cell especially, they differentiate into more antibody producing cells. So the lymph nodes become active at this time. So that's why they are enlarged. Okay, we will look at the structure of the lymph nodes in the next part of the lecture. Right. So the B cells, T cells, NK cells, when they are in contact with antigens from the organism, yeah, they will become the effector cells or the effector cells. Yeah, you can pronounce it either way. So the B cells will become plasma cells or antibody producing cells. Some of the B cells will become memory cells. Okay, memory cells. The T cells will become activated T cells, whereas the NK cells will become the activated NK cells. Okay. So these are all happening when or after they have been in contact with the organism. Who actually brought the antigen to them? This is the importance of the dendritic and the macrophage, because they are what we call antigen presenting cells. Okay, they will attack the organism, they will destroy, and then they will keep certain part of the organism, which we call antigen, and bring it to the T cells as well as the B cells for them to be activated. Okay, so that is in brief. Yeah. So this is the whole story with regards to the cells of the immune system. Again, to recap, from the bone marrow, they become the pluripotent hematopoietic stem cell with signals. They will be divided into the common myeloid progenitor cells or the common lymphoid progenitor cells. The myeloid progenitor cells become the granulocytes or the megakaryocyte progenitor. Okay, the megakaryocyte erythrocyte progenitors become the megakaryocytes, which then turns into the thrombocytes or the platelets. Some also will become erythroblasts and then mature into the erythrocytes. Okay, so this change into megakaryocyte and erythroblast take place in the bone marrow. You can see the bone marrow, blood, tissue, lymph nodes, and effector cells. Okay, as sites of where these events take place. I think we'll take 10 minutes break here. Okay. Okay.
Okay, Doctor. Sure.
All right. So let's resume the class. So I'll be recapping what I have just said previously. So we have the myeloid cells performing various functions in the immune response. Several types of myeloid cells include the macrophages and the neutrophils. These are very prominent, and they are the most prevalent in the body. And then we have the dendritic cells, equally important and have basically similar function to the macrophages. We have the third group, the secretory cells, because they have the ability to secrete or release granules. Yeah? So we have the eosinophils, And then most prevalent among all of these secretory cells, the neutrophils, and then the basophils. The last but not least is the mast cells. Very important, and we will see what the mast cells do later on. So let us look at the macrophage and the dendritic cells. Okay, I mentioned just now that these two cells, they have almost similar function. The major difference is if you look at the macrophage, they have also granules inside. Okay, the macrophage, they are cells that can do the phagocytosis. Phagocytosis means that they can engulf a pathogen or invading agent. They can also attack cells. Okay? Damaged cells, for example, can be destroyed by the macrophage through the phagocytosis. Once they engulf the invading pathogen, they can activate bactericidal mechanisms. These bactericidal mechanisms are carried out by the bactericidal chemicals that are present in their granules. Once these are degraded within the macrophage will maintain portions of the invading organism. They will then present what we call the antigen of the invading pathogens on their surface receptors. So they have an antigen presenting function. This is why we call the macrophage as an antigen presenting cells. The dendritic cells, they are also antigen presenting cells. Okay, they do the same kind of activity like the macrophage, but they have a longish, what we call longish and elongated cytoplasmic extension. Okay, unlike the macrophage is more globular, more spherical in their form. Okay, they are as mobile, and they could also extravasate. They can also move between the epithelial lining. Yeah, they can change their shape as well. But the dendritic cells, they have this very prominent elongation of the cytoplasm. Okay, we can't miss them, and this allows them to trap pathogens within an area in a more effective way. Okay, so they are very useful for antigen uptake in the peripheral sites. So we have talked about two antigen presenting cells. The rest of the cells, they do not have this antigen presenting capability. The neutrophils, for example, this is the most prevalent among the granulocytes. They can also engulf pathogens. So they have phagocytosis or phagocytic activity. And they have a few other specific functions. Okay, for example, they have the bactericidal mechanism, whether through the release within against the engulf antigen, or by basically explosion of themselves. Okay, perhaps I will show you in the next lecture of how the neutrophil can extravasate, okay, across the epithelial lining, and how they can also explode and then release their granules and kill the surrounding invading pathogens. The downside of the explosive capability of neutrophil is that when they explode, they become non-specific. They can also release and damage the surrounding host tissue, our own tissue. So they create a lot of inflammation. So the neutrophils, once they are activated, they uh, have to be controlled so that the inflammatory reaction does not go haywire, does not go too much. Yeah. So they need to initiate the inflammatory reaction, and once it's there, we have to then control so that it becomes advantageous to the body and not deleterious. The other granulocyte is the eosinophils. The eosinophils, unlike the neutrophil, you can see that the neutrophils, we can see the nucleus very prominent when we do the blood smear. We can see clearly the nodules of the neutrophils. The eosinophils nodule is more or less masked by the granules. Okay, we can still see the nucleus. They are mainly of 2 or 3 lobes, and they are masked. That is the appearance on the blood smear. And they are good for killing antibody-coated parasites. That is the function of the eosinophils. And then we have the basophils. Okay, basophils. The name suggests that this is because they can be detected or dyed with base dyes. The activated function of basophil is yet to be revealed, but they are thought to be important in cases of allergies. Okay, and basophils number in the body is never very high, so it's suggesting that their importance is not as prominent as the neutrophils and the eosinophils. One last group of cells is the mast cells. Okay, mast cells, they have a lot of granules as well, and most of these granules that the mast cells contain have serotonin and histamine. So very strong and very active substance. Again, they create a lot of inflammation around, and they are very prominent in cases of allergies and hyperactivities. So when we have asthmatic episode, for example, it is very common to see mast cells population shoot up and they increase in number. So it's one of the what we call the descriptive clinical feature of asthma. And when we take the blood sample, we see mast cell also increase in asthmatic episode. So this is the slide summarizing all type of the cells that we have mentioned. Now, this second big group of the immune cells is the lymphoid cells. Okay, lymphoid cells. They are mostly small and inactive when there is no infection. They are easily recognized because of the condensed chromatin in the nucleus and their relative absence of cytoplasm and more or less absence of the ER, yeah, endoplasmic reticulum. There are two major types of lymphoid cells, the B and T lymphocyte. I'm sure you have come across these two good friends of ours since your SPM. Yeah, so they are part of the adaptive immunity because they can be modulated. So they are good when we have we are defending against an evolving infection. So the B and T lymphocytes, they are adaptive in nature. Natural killer cells is also considered part of the lymphoid cells, but it is not part of the adaptive immunity. It's part of the innate immunity, which means that their its ability to change. Basically, there is a lack of ability to modulate, to change itself. So it's fixed. Yeah, unlike the B and the T lymphocyte, which can change their nature and their function. So this is what we mean when we say that the chromatin is very condensed. On this slide, you can see the blood smear on the left side. So lots of very red colored cells with a very pale central area. So these are the red blood cells, very easily recognized. Yeah, very easily recognized. So red blood cell is plentiful in the blood sample. You can see them clearly and easily. You cannot miss them. Big mistake if you can't recognize erythrocyte. Here we see one single solitary lymphocyte in the middle. Yeah, so you can see that the cytoplasm is almost absent. The whole cell is made up mainly of the nucleus, and the nucleus is you can see from the dye is very strong color, is because the chromatin is very condensed. The genetic material is very condensed. So it occupies almost the whole cell. Yeah? So we don't see the erythrocyte, we don't see the endoplasmic reticulum, and we just about able to see some part of the cytoplasm. So this is the feature of the lymphocyte that we commonly encounter in a blood smear. Okay. The natural killer cells, they also have granules. Okay, they also have granules. Their nucleus is also as condensed as the other lymphoid cells, but it is not as big. Yeah, it's not as big. We can see plenty of the cytoplasm. The defining feature is the lytic granules. They have granules that when they release will lyse the invading pathogen. And the main function of the NK cells is against viruses. Okay, not saying that they can't act against bacteria, but the body of evidence that we have now shows that the natural killer cells is very important when we have virus infection. Okay. I can take a few questions before we move on. If you have any question to ask, please turn on your microphone or write on the chat. Are you guys listening?
Yes.
Okay, so no question. Okay, so if there is no question, I will proceed to the next part of the presentation, which is on the tissues and organs of the lymphoid system. Okay, I hope you can see the slide well and my voice can be heard well as well.
Yes, doctor.
Okay, very good. Now, we have covered the cells of the immune system. Okay, now we are moving towards the tissues and organs of the lymphoid system. So the lymphoid organs, this is a term referring to the tissues containing large numbers of lymphocytes, okay, meaning that they have a lot of the B, T, and NK cells, okay, in a framework of non-lymphoid cells. And they are very important for lymphocyte development. Okay, as we will see, T cells, for example, they need to mature in these organs. They are also important for the initiation of adaptive immunity, because the macrophages and the dendritic cells, once they have become the antigen presenting cells, they will migrate to these lymphoid organs and present the antigens to the lymphoid cells for their stimulation. The lymphoid organs is also important because they provide the source for the tonic signals that is essential for the survival and maintenance of the cells, the lymphoid cells, while they are there. So they are divided into two. Firstly, we have the central or the primary lymph organs, and then we have the peripheral or the secondary lymph organs. This is the schematic diagram of the whole body, okay, showing the organs of the immune system, the adenoid, tonsil, the lymph node, okay, kidney also important, appendix, and then we have the lymphatics, and of course the bone marrow. We have also the large intestine and the small intestine, in particular the Peyer's patch, and then the spleen and the thoracic duct, where it provides the communication between the lymphatic system as well as the blood system. We have the thymus, and finally the left subclavian vein, also a point of communication. Okay, let us look at the central lymphoid organs. Okay, there are two central organs of the lymphoid system. Firstly is the bone marrow, and secondly the thymus. Okay, the bone marrow is the source of all the pluripotent hematopoietic stem cells. In particular to the B cells, this is also the place where the B cells will mature, okay, in the bone marrow. The T cells, on the other hand, will be released in their immature form, and they will reach the thymus, residing there until they become mature. The B cells and the T cells will both enter bloodstream after maturation. There will be some signals that stimulate them to be released from both the bone marrow and the thymus. And then they will migrate to the peripheral lymph organs. Okay? Of course, quite a number of them will also remain within bone marrow and thymus, but the majority will move to the peripheral lymph organs. So what are the peripheral lymph organs? What are they used for? These are very specialized areas in the body which function to trap antigens, also providing place where the immune cells can reside for temporarily while waiting for them to be initiated. Okay, so this is the place for initiating the adaptive immune system or adaptive immune responses. And they provide signals that sustain the recirculating lymphocytes. Examples are the lymph nodes, very important, yeah, and then the spleen and all the gut-associated antigen-bearing dendritic cells. Okay. This is a schematic diagram of the lymph node. It looks more or less like a kidney. Let's start with the right-hand side. Okay, you see the artery and also the vein. Okay, providing the circulation, the blood circulation towards the lymph node. Okay, so through the bloodstream, the immune cells as well as the pathogen and the antigen presenting cells will be brought to the lymph node. Okay, they will enter the lymph node. There are the afferent lymphatic vessels where the bloodstream will enter. This is the point where the bloodstream enter the lymph node, and then we have the afferent lymphatic vessel where the bloodstream leave, and then connect with the vein. So afferent and afferent. Afferent means or could be equals to the exit pathway. Okay, so when the bloodstream enters through the afferent lymphatic vessel, they will meet first the white area called the which is rich with the secondary lymphoid follicles. Okay, secondary lymphoid follicles. Okay, so this schematic diagram has been drawn to show the germinal center, the yellow part, okay, increase in size as we go down below. Yeah, so when there is no stimulation or initiation of the adaptive system, the germinal center remains very small. But as the B and T cells are being initiated and stimulated, they enlarge in size. Okay, they enlarge, and then once the infection is over, the germinal center can be reduced in size. They become senescent or become the old germinal center, and some of them will be basically destroyed by the system, and then we replenish the germinal with a new germinal center. Now, the blood stream carrying the antigen presenting cells will get in touch with the B and T lymphocytes in the secondary lymphoid follicle, and then move to the paracortical area. This area is mostly T cells. Again, more T cells will be recruited, and then we have the red area, okay, which is the medullary cords. Can you see my slide? Ada masalah tak? Tak ada. Okay, so the medullary cords in red, okay, and finally the medullary sinus, where the blood will be drained before entering the afferent lymphatic vessel. So as they drain out of the lymph node, they will also carry the activated B and T cells. Okay. Okay, somebody is entering the class. Okay. So I was talking to the class about the lymph node. Okay, and you need to be able to draw a schematic diagram of the lymph node, as well as to explain the flow of the events that takes place in the lymph node as the B and T lymphocytes are being activated in the adaptive immune response. Do you think you can do that? It's a very common final paper question, very likely to be used as part of the MCQs. It also very likely and very frequently used as part of the essay question. Okay, so you have to practice drawing as well as writing a very good and structured point by point essay for the events taking place in the lymph node. Okay. Right, the schematic diagram that we have seen is actually when we is a very crude way to represent the lymph node. Okay, but when we do the histology, it doesn't actually appear as such. Okay, I have to stop for 1 minute. Somebody is calling. Assalamualaikum. Yeah. Ya, betul. Apa dia? Ya, ya betul. Betul. Okey, boleh, boleh. Ya, boleh. Ya, boleh. Okey. Okey, Assalamualaikum. Sorry for the interruption. Okay. So I don't expect you to be able to draw this histology picture. Okay, just the schematic diagram. Okay. The next organ is the spleen. Okay, this is also very fairy tale like schematic diagram. The actual organ is much more complex. Okay. In the next slide, this is also an attempt, a schematic diagram of the spleen to allow us to understand better the spleen. Okay, so if we look at this slide, look at the transverse section of the white pulp on the left and the longitudinal section of the white pulp. Okay, so the spleen is actually an organ which contains two major parts, the red pulp and the white pulp. So we are talking about the white pulp here. Okay, so in the middle we have the germinal center. Okay, the germinal center. Again, the germinal center is small when there is no action and it enlarged when there is a lot of activities. So this is the central point in the organs, the germinal center. In the spleen, the germinal center has an area crowning it. So we call that area the corona. Yeah, corona means crowning. Yeah. So corona is not a very new name. Yeah, we have been using corona for many things in biology. So this corona section, it appears as the yellow area in the slide, in the diagram. This is rich in the B cells. Okay, rich in the B cells. And then we have also the pulse, yeah, the pre-arteriolar section, okay, the pulse which contain mostly the T cells. Okay, within the pulse there is the central arteriole where the blood is taken in. The blood cells containing the macrophage and the dendritic cells, the antigen presenting cells, then can activate the T cells as well as the B cells, yeah, within the pulse and the germinal center. Okay. So, and finally we have the marginal zone, which is the zone between the white pulp and also the red pulp. So the spleen has many other functions, especially with regards to the hematology, but this is the one that we are concentrating on here is on the contribution or the role of the spleen to the immune system. Now, if you look at the spleen on the histology slide, it will appear something like this. Yeah, the germinal center is in the middle, GC, and then the crown or the B cell rich area, the corona area, and then we have the pulse, yeah, the periarteriolar lymphoid sheath containing a lot of the T cells, and then we have the marginal zone surrounding this area, and then the perifollicular zone. Again, another layer before we reach the red pulp. Okay. So that this is a slide that summarizes the previous two slides. Okay, so we have talked about the thymus and the bone marrow. Sorry, the spleen and the lymph node. Sorry. Now we come to the another peripheral lymphoid organs. Very important one as well, the gut-associated lymph tissues. They are very important, although they are the peripheral one. Okay, we have lots of materials coming into our gastrointestinal tract, food, bacteria, viruses, all sort of things through our food, through our fluids, and they can invade the GI tract at any time. So the gut-associated lymphoid tissues include the tonsils, the adenoids, appendix, and also Peyer's patches. They collect antigens from epithelial surfaces of the GI tract. with the pairs, patches being the most organized and the most important. The pairs, patches, they have specialized epithelial cells, sorry for the typo, very specialized epithelial cells called the multi-fenestrated or the M cells. These M cells have a large central dome of B cells surrounded by the T cells. Okay, this is a schematic diagram of the Peyer's patch. Okay, so we have the germinal center in the middle, and then it has a dome. Okay, and then the follicle containing the B cells as well as the T cells area. So they are beneath the epithelial cells. Whenever there is a breach here, they will detect the invading agent. Okay, I would like to show to you the video of the gut-associated lymph tissue. I hope we can watch it together. Okay, please tell me if you can see the video. Nampak ke?
Tak
No.
nampak.
Tak.
Sorry,
Dengar je.
I cannot see.
you cannot see. Okay. Sekejap saya kena stop dulu kot. I will stop presenting the slide and I will show you the. Tak ada. Kan. Macam mana? Okay, maybe here. Can you see? Boleh nampak ke?
Yes.
Okay, so this is showing the it says immunology and the gut mucosa. So let's have a look at this video.
Yes.
The human gut can be the scene for devastating conditions such as inflammatory bowel disease, which arises through an improperly controlled immune response. The gut is often the body's first point of contact with microbes. Every mouthful of food is accompanied by a cargo of microorganisms that go on to encounter the mucosa, the innermost layer of the gut. Most microbes are destroyed by the harsh acidic environment in the stomach, but a hardy few make it through to the intestine. The intestinal surface is covered with finger-like protrusions called villi, whose primary function is the absorption of nutrients. However, these structures and the underlying tissues also host the body's largest population of immune cells. Scattered along the intestinal mucosa are dome-like structures called Peyer's patches. These are enriched in lymphoid tissue, making them key sites for coordinating immune responses to pathogens whilst promoting tolerance to harmless microbes and food. The villi contain a network of blood vessels to transport nutrients from food to the rest of the body. Lymphatics from both the Peyer's patches and villi drain into the mesenteric lymph node. Within the villi is a network of loose connective tissue called the lamina propria, and at the base of the villi are the crypts, which host stem cells that replenish the epithelium. Finally, the epithelium, together with its thick overlying mucus, forms an important barrier against microbial invasion. Embedded within the matrix of the Peyer's patch is a mix of immune cells, including T and B lymphocytes, macrophages, and dendritic cells. A key function of the Peyer's patch is the sampling of antigens, in this case mostly bacteria and bits of food. To facilitate this, the Peyer's patch has a much thinner mucus layer as well as specialized phagocytic cells called M cells, which can transport material across the epithelial barrier via a process called transcytosis. Finally, dendritic cells are able to extend dendrites between epithelial cells to sample antigens that are then broken down and used for presenting to lymphocytes. Sampling antigens in this way typically results in so-called tolerogenic activation, where the immune system initiates an anti-inflammatory response. With their cargo of antigen, these dendritic cells then traffic to the T cell zones of the Peyer's patch. Upon encounter with specific T cells, the dendritic cells convert them into an immunomodulatory cell called a regulatory T cell or Treg. Defects in the function of these cells are associated with inflammatory bowel disease in both animals and humans. These Tregs then migrate to the lamina propria of the villi via the lymphatics. Here, the Tregs secrete a molecule called IL-10, which exerts a suppressive action on immune cells within the lamina propria and upon the epithelial layer itself. IL-10 is therefore critical in maintaining immune quiescence and preventing unnecessary inflammation. However, a breakdown in this process of immune homeostasis results in gut pathology, and when this occurs over a prolonged period and in an uncontrolled manner, it can lead to inflammatory bowel disease. Chemical, mechanical, or pathogen-triggered barrier disruption, coupled with particular genetic susceptibilities, may all combine to set off inflammation. Epithelium coming into contact with bacteria is activated, leading to bacterial influx. Alarm molecules released by the epithelium activate immune cells, and Tregs in the vicinity scale down their IL-10 secretion to enable an immune response to proceed. Dendritic cells are also activated by this environment and start to release key inflammatory molecules such as IL-6, IL-12, and IL-23. Effective T cells also appear on the scene, and these coordinate escalation of the immune response by secreting their own inflammatory molecules, tumor necrosis factor alpha, interferon gamma, and IL-17. Soon after the effector T cells have arrived, a voracious phagocyte called a neutrophil is recruited. Neutrophils are critical for the clearance of bacteria. One weapon in the neutrophil armory is the ability to undergo a dramatic form of self-destruction called mitosis. This leaves behind a jumble of DNA saturated with enzymes called a neutrophil extracellular trap. Although this can effectively destroy bacterial invaders and plug any breaches in the epithelial wall, it also causes collateral damage to tissues. Slowly the tide begins to turn, and the bacterial invasion is repulsed. Any remaining neutrophils die off via apoptosis, a non-inflammatory form of cell death, and are cleared by macrophages. Epithelial integrity is restored by replacement of any damaged cells with new ones from the intestinal crypts. Finally, Tregs are recruited once again to calm the immune response. Targeting the molecules involved in gut pathology is leading to effective therapies for inflammatory bowel disease.
Okay, so I hope you enjoyed the video. It's pretty much summarize what we have talked about the function of macrophages, dendritic cells, and it also has a very good presentation on the explosion that took place once the neutrophil is being activated, and the collateral damage that occur to the surrounding tissues. Very good video. So this video also talk about the gut-associated lymph tissues. The same kind of reaction take place elsewhere, okay, in the peripheral tissues, and you can revisit the video by clicking. There is a link that I've put at the title. You can click on the title, control, and then left click, so you can revisit the video. And I would also like to recommend you to spare some time and have a look at all the other videos that are linked to this video. Yeah, you can see on YouTube there are plenty of very good representative videos that have been prepared and which would be very helpful for you to understand what immunology is all about. Okay, so the Bayes patch on histology will appear something like this. Yeah, the M are all the fenestrated area. Okay, I think we are at the last part of today's lecture, which is on the circulation between the blood and the lymphatic system. So the naive lymphocytes, okay, before they are being activated, they are already mature, but they do not they are not yet active. So we call them naive lymphocytes. They circulate continually from the blood into the peripheral lymphoid tissues, and they enter by squeezing between the specialized endothelial cells. These cells are called the high endothelial venules. And then they can return back to blood by the lymphatic vessels or directly into the blood in the spleen. So there are a few communication points between the lymphatic system as well as the blood system. And whenever there is infection occurring, they become arrested in the lymphoid tissues, being activated, and then become the effector cells. Okay, so the T cells, as you see in the video, they turn into the Treg. You will come across later on a few more types of effector T cells, the cytotoxic T cells, the helper T cell, etc. There are a couple of types. The B cells, on the other hand, when they become effective, they become active, they are stimulated, they turn into the plasma cells as well as the memory cells. The plasma cells produce antibodies, and these will be phased out once infection is over. The memory of the infection will be retained in the memory B cells. So some numbers of B cells, not that many, a few, will be retained, will be maintained at some level within the body at all period of time. So when there is a second succeeding infection, then this memory cell will be reactivated immediately, and then the specific antibodies can be produced rapidly. So the last slide is summarizing again the circulation that takes place in the immune system. Yeah? So if there is an infection in the peripheral tissues, the antigen will be brought to the lymph nodes, okay, and then the immune cell will be activated. Some will go to the blood circulation, and then the circulation then flow between the lymphatic vessels and the blood vessels. Okay, there are several points of contact like the thoracic duct, etc. I guess that is the last slide. If you have any question before we end the class, please fire away.
No, Doctor.
Okay. If there is no question, I guess it's not very difficult. Yeah, it's not a difficult class. It's not a difficult part of the immunology. You have heard it before. Yeah, it's not difficult at all. I have given to Farah the list of topics to be chosen. Please arrange yourself into group of 5, and then each group pick one topic. Yeah. So there should not be any overlap because I've already sent out 8 topics. You need to discuss on how to then present this online live. Yeah, I don't want a pre-recorded one. I would like you to present it live on the date that I have specified. Yeah, I hope that you will arrange it in such a way that no other classes will take place at that time. Okay. And there will be 5 minutes question and answers session after the 10 minutes presentation. I think that's all. Yeah, please make sure that you sign up for the you time in for this class as well. If there is no question, that's all. We will see you again next week around the same time. Wassallallahu ala sayyidina Muhammad wa ala alihi wa sahbihi wa sallim walhamdulillahi rabbil alamin. Okay, thank you very much.
Thank you, Doctor.
Thank you, Doctor.